So, students, our next topic is about the prokaryotic cell. Under the prokaryotic cell comes the bacteria, all kind of bacteria, blue-green algae or cyanobacteria, nitrogen-fixing bacteria. Okay, so that means all Monera. All Monera kingdom comes under this one. Mycoplasma, PPLO. Now, talking about the structure of a Monera, if I talk about a bacteria, suppose this is a bacteria, the bacteria, in their structure, the outermost layer, there is a cell wall. There is a cell wall which may be sometimes thick and sometimes a little thin. So this is a cell wall, and this cell wall, it is made up of peptidoglycan. Peptidoglycan is a compound which is made up of N-acetyl muramic acid and N-acetyl glucosamine. N-acetyl glucosamine. Now, the presence and absence of this peptidoglycan will decide if it is a gram-positive or a gram-negative bacteria, in which the peptidoglycan is the cell wall in which the peptidoglycan is around 90% of its composition are gram-positive. Are gram-positive bacteria, and those bacteria in which the cell wall has only 10% of peptidoglycan, they are called as gram-negative. Though this is not the exact reason of calling them as gram-positive or gram-negative, they are called as gram-positive or negative because by their ability to accept the stains, that gram stain. If a bacterial cell is able to get stained by grams stain, then it will be called as gram positive, and otherwise it will be called as gram negative bacteria. So it has been observed that those who have more peptidoglycan, they are able to take up more stain, and then they will be called as gram positive, and those who are not able to take up the stain, they have less peptidoglycan in their cell wall. Then outer to this cell wall, it is also observed that they will have either slimy layer, sometimes they will have slimy layer around, or sometimes they will have capsule. If it is a slimy layer, then it will be more loose kind of a structure around, and if it is a capsule, which will be a little more rigid and hard structure. Then inside the cell wall, there will be plasma membrane. The plasma membrane composition is similar to that of the eukaryotes, having lipid bilayer. So the outer layer is slimy layer, or it can be a capsule. And innermost one, plasma membrane. The plasma membrane, in order to increase its surface area, it has got various infoldings like this. These infoldings, they are called as mesosome. Or they are also called as chondrioids. The reason behind calling them as chondrioids is that they will do the function of mitochondria. So the name chondrioids. So they will be helping in making ATP molecules and respiration. That's why they are called as chondrioids. They also help in attachment of the DNA or the genopore. So in this cell, there will be a single circular DNA. Suppose this is the DNA. This is a single circular DNA which will be covered by a lot of proteins. And this DNA will be attached to this mesosome. This mesosome's function is also that it will help in replication. It will also help in secretion of lot of enzymes. Then other than that, the bacteria also has got lot of chromatophores in it. For example, in blue-green algae, they have chromatophore pigments. The bacteria also has got cilia, flagella, and pili and fimbriae on its outer structure. If it is a flagella, we will observe that it will have a basal body and then a hook. This is the basal body and then this is the hook, and from which a filament will arise, and this becomes this whole structure, it will be called as a flagella. Flagella in bacteria is also very complicated structure. We will discuss about flagella structure when we discuss the eukaryotic cells. The ribosomes in the bacteria can be attached to the plasma membrane, or it can be freely floating also. These ribosomes, they are of 70S type. So we know that in ribosomes there are two subunits, and here the subunits are 50S and 30S type. Here S means the Swedberg's unit. S stands for Swedberg's unit. That means it will decide what is the weight of that substance, weight of that unit. Then, as I have told you, there can be flagella, there can be some small hair-like structures called as pili. Pili will be responsible for attachment of one bacteria to another bacteria during sexual reproduction or during conjugation. Sexual reproduction. And there are some hair-like or bristle-like structures. These are called as fimbriae. And these are helping the cell in attaching to another cell during the time of infection. So if the cell wants to infect another cell or it wants to attach itself to a host, then fimbriae is required. Attached to host. So other than that, the cytoplasm contains all the raw materials that are required to produce different components in the cell. So this is all about the prokaryotic cell.